Hello, this is Guite from Department of Botany to Champu College. In this video lecture, I'm going to discuss about the second part of a two-part series. In the first part, I describe secondary and anomalous secondary growth in plants in a nutshell. In this, I'm going to go more in depth into that and also talk about how aspect of anomalous secondary growth in Amaranthus, Mirabilis and Dracaena species with the help of diagram. I hope you all know the difference between normal secondary growth and anomalous secondary growth in plants, which we already discussed in the previous slide. So here, based on a Manipur University syllabus, I'm going to discuss about anomalous secondary growth in three plant species, that is Amaranthus species, Mirabilis species and Dracaena species. This Amaranthus, Mirabilis, they were under a dicot plant. All right, dicot plant is under angiosperm. When we talk about angiosperm, then it is a flowering plants and it will produce seeds. And this Dracaena is also under angiosperm. Even though it is angiosperm, it will be included under monocot. That means monocot plants also they produce flower, but they'll be having one cotyledon. That means in case of grass family or in case of maize, but here we'll be studying in case of Dracaena. So this is a normal uh, tissue representing the secondary growth. All right, so here let's see for Amaranthus. Anomalous secondary thickening is due to the formation of accessory cambium. Abnormal activities of the abnormal cambium. Here new cambial rings are formed from the cortex or pericycle. After the order cambium have ceased, that means stop its functioning, the new cambial strip form also behave abnormally. So in case of Amaranthus, how the anomalous secondary growth is produced because of the activities of abnormal activities of cambium. That means they'll be having extra cambium. In a normal secondary growth, you'll be having cambium below the pericycle. All right, so let's assume this is a normal secondary growth or normal vascular bundles. So these are the vascular bundles, and the vascular bundles will be arranged in a ring-like structure. But in case of Amaranthus, they will be having accessories, that mean extra cambium, and that extra cambium will be abnormal. All right, it will be not normal. Mostly in a normal secondary growth, cambiums are produced in the below the pericycle. All right, below the pericycle. But since it is already abnormal, the cambiums or the secondary vascular bundles will be produced from the pericycle. All right, pericycle means the tissue which cover the secondary vascular bundle or normal secondary growth in case of plant. But since it is abnormal, cambium they can be produced from the pericycle or they can be produced from the cortex. And even though they produce a cambium, the mode of activities of cambium will be abnormal. And in case of mirabilis also, the mode of activities or the place where cambiums will be produced will be from pericycle again, and the activities of cambium will be a bit uh, just a little bit difference between the Amaranthus and the mirabilis. And during exam, if you memorize one, that means if you know the secondary growth for Amaranthus or in case of mirabilis, you can write at the same point, but there is only a slight difference, and that is negligible. So if you know the definition of or the description of Amaranthus anomalous secondary growth, you can use it for mirabilis also during exam. And question number 2, anomalous secondary growth in monocotyledon plants in Dracaena. So here normally a monocot, all right, majority of the monocot plants they do not produce cambium and they have xylem and phloem. But in case of this Dracaena, even though it is a monocot plant, they have a what you call that is cambium. Because of having cambium, they can produce a secondary growth. All right, since they have a cambium and produce a secondary growth, it is already anomalous because it is deviating from the normal monocot plants. All right, because of that one, it is called anomalous secondary growth in Dracaena. These are some pictures of plants under Amaranthus genus. Keep in mind that under Amaranthus, different types of species will be present, because this Amaranthus, it is a genus. And in a local dialect, some species including under Amaranthus like the Bongekte, Jauzamte and Huangte are under Amaranthus genus. And in the next slide, we'll be cutting this stem in a transverse section, alright, in a transverse section, and we'll be observing what are the anomalous characters taking place inside this stem. From now on, I'll be talking in details how does the anomalous secondary growth occur in case of Amaranthus. And this is a picture representing anomalous secondary growth in case of Amaranthus, and below is a picture representing anomalous secondary growth in case of a normal plant. I have put this picture just to compare the difference between anomalous secondary growth and normal secondary growths. Point number one, the normal rings of vascular cambium, that is primary vascular cambium, is absent in the Amaranthus stem. So in case of Amaranthus, the primary cambium or the primary vascular cambium, they will not arrange in a ring-like structure. Even though primary vascular cambiums were present, they will be scattered here and there. Alright, they will not form a ring-like structure. In a normal vascular bundles or the cambium, they arrange in a ring-like structure. If you see here, in case of this one, they will be arranged in a ring-like structure. But here in Amaranthus, it will be scattered here and there inside the pit. Alright. Number two, anomalous secondary growth take place due to the development of new extrastellar meristem or accessory cambium. That is the cambium outside the stele in the pericycle region. The meaning is that the anomalous secondary growth occurs due to the activities of some extra cambium. That is, accessory cambium will be formed. In a normal secondary growth, cambiums will be produced below the pericycle, but in case of this Amaranthus, cambium will be produced in the pericycle region, in this region. Alright, actually in a normal secondary growth, cambium is produced below pericycle, but in case of Amaranthus, cambium will be produced from the pericycle, that is the extra cambium. Alright, extra cambium will be produced from pericycle. A layer of cells from the pericycle become active. These pericycle cells will become active and form the cambial ring. That is, they'll form a ring-like structure. Here, they'll form a ring-like structure. This one, this is a cambium. That is secondary ring. The extracellular cambium behave abnormally. This cambium, which is from the pericycle region, the mode of activities of cambium, it will be totally different from normal secondary growth. So next, initially it cuts small amounts of parenchyma on the outer side and then start to cut alternating patches of secondary xylem and parenchyma internally. So the cambium, the secondary cambium, which is from the pericycle, at first it will cut small amount of parenchyma. This is a cambium, small amount of parenchyma on the outer side. So small amount of parenchyma on the outer side, and then start to cut alternating patches of secondary xylem and parenchyma internally. So after this one, they will cut secondary xylem. Alright, this is the secondary xylem. That means alternating with what? Patches of secondary xylem and parenchyma internally, and somewhere here also they will produce some amount of parenchyma. Toward this outer side, they will cut what? The secondary phloem opposite to secondary xylem. This is how secondary phloem is produced, and parenchyma above the secondary parenchyma, and somewhere a parenchyma will be produced, and after that one another layer of parenchyma will be produced, that is called secondary parenchyma. This results in the formation of conjoined collateral secondary vascular bundles arranged as a ring, and it appears as embedded in the parenchymatous ground. So here cambium present in the middle, and after that one secondary phloem, below secondary cambium, secondary xylems are present. So this cambium conjoined means xylem, cambium and phloem were present in one bundle. Alright, collateral, and arranged in a ring. They will appear in a ring-like structure. This will form in the ring. This is This picture is a magnified version of some portion of Amaranthus dicot stem. So, and it appears as embedded in the parenchymatous ground. Parenchymatous grounds then mean the cells or the tissue which surround the vascular bundle. Somewhere here, they were called parenchymatous ground. That means it looked like that these vascular bundles were arranged in a parenchymatous tissue. After some time, the cambium become inactive. So after some time, this cambium will become inactive and stop its functioning. However, secondary accessory cambium develop from the secondary parenchymatous cells produced by the first extracellular cambium. So after some time, this cambium will stop its activities, and from another parenchymatous cells, another cambium, another layer of cambium will be produced. All right, that is how the mode of activities of Amaranthus anomalous secondary growths occur. The newly formed accessories cambium also behave like the previous one. Thus, the secondary vascular bundle ring alternate with the first one. So, as we already discussed before, the mode of activities of cambium will be first they will produce xylem. Another layer of cambium will be there. There will be another layer of cambium. They will produce xylem, parenchymatous, and after that one, secondary phloem and parenchymatous will be producing. And that cambium will stop their activities again, and another layer of cambium, secondary layer of cambium will be produced, and because of that one they will form a ring like structure. All right, the newly formed accessory cambia also behaves just like the previous one. Thus the second vascular bundle ring alternate with the first form one. So here if you see, this is the first and this is the newly one, this is the second one. If you see here, the mode of arrangement of this vascular bundle 1, vascular bundle 2, they arrange in alternate, that means side by side. All right, that is the meaning. The second vascular bundle ring alternate with the first one. Second vascular bundle alternate with the first form one. So subsequently, more and more cambions are produced, and all of them behave exactly as mentioned above. Thus, a number of concentric rings of vascular bundles which appear as scattered in the ground tissue. So, because of activities of many cambium, numbers of concentric ring. Concentric mean different circle. All right, yeah, one circle, another circle. So another circle ring, concentric rings will be produced. The last cambium ring behave differently. It cuts secondary xylem alternating with sclerenchyma on the inner side. So one last activities of cambium will be present here, somewhere here. So the mode of activities of this cambium is totally different from that the first, second, third, and the another activities of cambium. Here it cuts secondary xylem alternating with, that means they will cut secondary xylem below, and after that one sclerenchyma. All right, sclerenchyma mostly they were tissues. All right, hence the mostly they were dead tissue. Hence the last ring of vascular bundle appeared to be embedded in the sclerenchymatous zone. So since the last cambium produce secondary xylem alternating with sclerenchyma, after secondary xylem sclerenchyma, and the last cambial ring, this portion, the last one, appear to be embedded in a sclerenchymatous zone. All right, as if that the vascular cambium were arranged in arranged under sclerenchymatous tissue. In the mature stem, the two medullary bundles and the pit disintegrate and form a cavity. So in a mature uh, bundles, so when the plants attain maturity, two medullary bundles. All right, medullary bundles somewhere here, one, let me write it. Suppose this is one and another layer, this is two. So in the mature stem, the two medullary bundle, let's say this is one medullary bundle and this is another one medullary bundle and this is another medullary bundle, and the pit disintegrate and form a cavity. This one, it will be disintegrated, I mean it will be uh, broken down and they'll form a cavity. All right, they'll form a this portion is a cavity, and this is how it is formed. Here we'll be discussing in details how does the anomalous secondary growth occur in case of mirabilis. This is a mirabilis xylapa plant. Another term of this mirabilis xylapa that is 4 o'clock plant. All right, 4 o'clock plant. So how did they got their name? That is 4 o'clock plant, because they will open their flower late in the evening. All right, suppose if this one is closed, but they'll open this flower late in the evening around 4 p.m. and they remain open till the next day morning around 4 a.m. in the morning. Because of that one, it is also known as 4 o'clock plant, because it remains open overnight and close in the early morning. And sometimes they will open their flower during a cloudy day. All right, the secondary thickening in mirabilis also due to the formation of accessory cambium from external to the primary vascular bundle. Here also the secondary thickening is due to extra cambium, that is outside the normal, that is the primary vascular bundles. More or less, if you know the secondary or anomalous secondary growth in case of Amaranthus, during exam you can just write either of them. Mirabilis stem belong to the family Nyctogenaceae. Anomalous secondary thickening. The section is rather quadrangular in outline. If you look at here, this is the STS of Mirabilis jalapa. It looks like it have 4 quadrangular, that means 4 sides, 1, 2, 3, 4. The anomalous secondary thickening is more or less similar to that of the Amaranthus stem. So the anomalous activities of this Amaranthus and this Mirabilis, it is similar. During exam also, not 100% similar, but there is more similarity compared to another species. The extracellular cambium is formed from the pericycle. So pericycle, from the pericycle, cambium will be produced. If you don't know where is pericycle is located, go back to Amaranthus. I already explained in details. This extracellular cambium, this cambium, alright, cut internally in the inside secondary xylem. It will cut secondary xylem with somewhere here parenchyma, and above they will cut secondary phloem, and after that one they will cut secondary parenchyma and some parenchyma they will cut somewhere here. That means this vascular bundle is cambium, secondary xylem and secondary phloem were surrounded by parenchymatous tissue or parenchymatous cells. Thus, rings of conjoined collateral vascular bundles is formed, which is separated by the parenchymatous cells. So this is one bundle, and this is another bundle. They will form a ring-like structure. This is a vascular bundle. They will be arranged in a ring-like structure. They will be arranged in the ring-like structure. This is what they mean, collateral. All right, vascular cambium or vascular bundle. Upper side phloem, lower side xylem, and the middle will be cambium. The first form cambium ceases its function after the formation of vascular bundle. After this formation of complete vascular bundle, this cambium, they will stop its function, and somewhere above this cambium another cambium will be produced again from the parenchyma. All right, from the parenchyma, that is secondary parenchyma. Successive rings of accessory cambia are formed externally from the secondary parenchyma. So this cambium already died the first ring. They form a ring-like structure. They will die. After they die, just above the secondary phloem, secondary parenchyma will be produced. The secondary parenchyma will produce another layer of cambium. And when they produce another layer of cambium again, they'll be producing secondary xylem and secondary phloem again. And then one will die again. And after that one, they'll form another cambium again. So because of that one successive ring, more than one types of rings will be produced. Let's assume this is the first, second, third, and this ring they will be having vascular bundle. That means xylem and phloem will be present. All right, in this ring. So that types of rings having more than one ring, they will call concentric ring, which will be present inside the stem. Next, the last ring cuts secondary xylem alternating with sclerenchymatous cells internally and secondary phloem alternating with parenchymatous cells externally. Thus, the last ring of vascular bundle is embedded in the sclerenchymatous cells. Let's assume that this portion is the last ring. Alright, this let's assume this portion is the last ring. This is a cambium. The last ring will cut secondary xylem alternating with sclerenchyma gametas. After secondary xylem, they will cut sclerenchyma gametas cells. That means sclerenchyma gametas cells are those cells that when they become maturity, they tend dead. Alright, they become dead. They become dead when they attain maturity. And above this cambium, they will cut again secondary phloem together with parenchyma gametas cells. So since there is sclerenchyma gametas cells together with secondary xylem along with cambium and along with secondary phloem, it looks like that this bundle were arranged inside the sclerenchyma gametas cells. The medullary bundles are more numerous. So if you see here, this is a medullary bundles, the small one. These bundles were more in number compared to the inner side. Those according to what periphery are smaller in size and more numerous. That means these medullary bundles, which occur in the outside, they were more in number at the same time. They were smaller in size compared to the bigger one. Whereas those at the center are larger and more scattered. They are more scattered. That means these the bundles, vascular bundles, which are present in the middles, they were bigger in size. At the same time, they were more scattered compared to the smaller one. That is a medullary bundle. Anomalous secondary growth in Dracaena. That is a monocotyledon plant. According to a MUR syllabus, this Dracaena is the only monocot plants given by them. So during exam, they may not mention the name Dracaena. Instead, they can ask you write the anomalous secondary growth in monocotyledon plants. During the time also, you need to keep in mind that it is Dracaena. Let's see, these are some pictures which represent Dracaena plant, Dracaena species. This is another type of Dracaena species. Alright, Dracaena, it is a genus. A monocot Dracaena is a typical example of anomalous secondary thickening in monocots. Typically, secondary thickening is absent in monocots. Therefore, secondary thickening itself is an anomaly, as Dracaena is a monocot. The secondary growth occurs in herbaceous and woody Liliaceae family, Dracaena, Aloe, Yucca, and other groups of monocots. The meristematic concern with this growth is called cambium. So the activities of anomalous secondary growth in case of this Dracaena is because of cambium. Alright, majority of the monocotyledon plants, that is monocot plants, they do not have cambium. Since they do not have cambium, there is no secondary growth. Alright, so this Dracaena, it is deviating from the normal secondary growth or deviating from the normal monocotyledon plants, because majority of the monocot plants, they do not have cambium and they do not produce secondary growth. Since they have cambium, they produce secondary growth, and that type of secondary growth which is produced in monocotyledon plants in case of Dracaena, it is called anomalous secondary growth because it is abnormal compared to normal secondary growth. Mostly it is common in Liliaceae family in case of Dracaena. Then in case of aloe vera also, they have cambium. Alright, it is a monocot. That means the seeds, you will not be able to divide into two different cotyledon. Alright, only one cotyledon. So let's see. This is a picture or diagram representing anomalous secondary growth in Dracaena. So these are normal secondary growth. Alright, they are called primary vascular bundles. It includes ground tissue, that means parenchyma tissue, it will be included. And from here to here, that means from here, these are the anomalous secondary growth. Alright, anomalous secondary growth in case of Dracaena. Let's see. In the region which have ceased to elongate, some cells occurring outside the vascular bundle. Some cells were occurring outside the normal vascular bundle. That means this portion is outside. Become meristematic. That means they can become a dividing cells and form the what? They will form the cambium. This portion will form the cambium. The secondary meristems originate in the cortex. In fact, deep layers of cortex or pericycle. That means the secondary meristem, this whole tissue originate in the where in the cortex. Cortex in which side? Deep layers of cortex. So if you go down from here, that means deep layer of the cortex. Here we have a cambium. Alright, or in the upper side of the pericycle, somewhere here pericycle will be present. Pericycle, that's the meaning. And the cambium cells are fusiform or rectangular in shape. If you see here the cambium, this is a cambium, they are fusiform or rectangular in shape. Next, instead of forming phloem and xylem on the outer and inner side as in normal condition, the cambium cells goes on dividing and producing secondary tissue, xylem, cambium and phloem on the inner side first. So if you see in case of secondary growth in Mirabilis and Dracaena, these cambium cells in the outside they will produce phloem, and in the lower side they will produce xylem and parenchymatous, phloem, and in the outside phloem and secondary parenchyma. But here all the vascular bundles will be produced below or down the cambium. That means the cambium cells goes on producing secondary tissue on the inner side. Then when they will produce, this cambium cell will produce xylem and phloem in the inner sides of the cambium, then below the cambium. And after that one, in the upper side they will produce small amount of parenchyma. Alright, that means what? Instead of forming phloem and xylem on the outer and inner side as in normal condition. In a normal condition, xylem and xylem will be produced below and phloem will be produced above the cambium. But here it is not produced. Both the xylem and phloem bundles were produced below the cambium. The cambium cells goes on dividing and produce secondary tissue, that is xylem and phloem on the inner side first. And after that one, they will produce xylem and phloem. They are called secondary vascular bundle. Alright, they produce small amount of new tissue are cut off, then will produce on the outer side as well. This is the new tissues are parenchymatous. So that's it. Here, those formed on the inner side differentiated into oval shaped vascular bundles and radially arranged parenchyma cells. If you see here, those vascular bundle, alright, the secondary vascular bundle which are formed below the cambium, they look like oval in shape. This oval, that means X shape. Alright, and radially arranged, that means they will be arranging side by side with the parenchyma cells. Alright, here somewhere here, this were parenchyma cells. These parenchyma cells in which the vascular bundle remain embedded are said to constitute the conjunctive tissue. Those parenchyma cells in which vascular bundle, this one or this portion will be the parenchyma cells, white and the black color, it will be the vascular bundle. Vascular bundle present inside the or together with the parenchyma cells, they will call conjunctive tissue. I hope it will be clear. What is conjunctive tissue? Those vascular bundle embedded within the parenchymatous tissue below the cambium, they are called conjunctive tissue. The secondary bundles are mostly amphiβasal. That means secondary vascular bundles are amphiβasal. That means xylem will be surrounding phloem. So here we have phloem bundle, and the outer side it will be xylem. Alright, that means xylem will be surrounding the phloem bundles. So here also let's imagine the upper side, outside, that is xylem, and the inner side it will be phloem. That type of vascular bundles are called amphiβasal arrangement, and some of them may be collateral as well. Collateral means those xylem and phloem which are present in one bundle, they will call collateral vascular bundles. Alright, collateral vascular bundle. And xylem and phloem present together, they will call collateral vascular bundles. The tissue cut off by the cambial cells on the outer sides are scanty in amount and are parenchymatous in nature. That means the tissue which are cut off by the cambium in the outer side, they are small in amount, scanty, small in amount, and are parenchymatous. That means they will be producing lots of chloroplast, and more or less they will be green in color for storage. That is the characters of parenchymatous. The primary bundles are comparatively larger. These are primary vascular bundles. They are comparatively larger compared to the secondary vascular bundle. They are also mostly amphiβasal or rather rarely collateral one. That means they are also mostly what? Amphiβasal. That means the vascular bundle phloem will be surrounded by xylem bundle. That is the meaning. And they are collateral ones. Collateral ones mean xylem and phloem. Alright, they'll be present in one bundle. Alright, or phloem and xylem will be present in one bundle together. They are called co-literal ones. I have completed a topic on anomalous secondary growth in plant, that is in case of Amaranthus, Mirabilis and Dracaena. So this Amaranthus and Mirabilis, it is a dicot plant, and this Dracaena, it is a monocot plant. Now let us analyze a previous year question from Manipur University which I have collected. And during exam, if they happen to ask you again, you need to draw the diagram first, and after that one description, the wording. All right, so this let's see question number 1. State anomalous secondary growth in Amaranthus and Dracaena stem. It was asked in the year 2012. And describe anomalous secondary growth in Mirabilis stem. It was asked in the year 2014. Describe anomalous secondary growth in Mirabilis stem. 2016. Describe how anomalous secondary growth take place in Amaranthus. It was asked in the year 2018. As I already mentioned, diagram it is to carry 2 marks, and description it will carry 4 marks. That means total 6 marks. Diagram is very important, and if they happen to ask you again in the next exam, memorize nicely. It's not difficult. Diagram and all the description, and you need to practice how to draw the diagram, memorize each and every parts of the stem or the tissue. I hope that's helpful.